Hi everyone, my name is Alex, and the title of my project is Possible Use of microRNA 488 as a Biomarker for Bad Prognosis in Type 2 Diabetes. Type 2 diabetes is a serious public health issue. It affects approximately 462 million people worldwide, which is 6.28% of the global population. In Hawaii, 9% of residents age 18 or older have type 2 diabetes. Furthermore, of that 9%, 13.1% are native Hawaiians and 23% are other Pacific Islanders, indicating a high degree of health disparity. Type 2 diabetes is a chronic disease that affects the body's processing of glucose. When we eat food, such as carbohydrates, our stomach processes that into glucose molecules. Those glucose molecules will then enter the bloodstream where cells can uptake it and use it to convert into energy. However, in order for cells to uptake the glucose, the hormone insulin is required. Insulin is a hormone that is secreted by the pancreas and activates a signal transduction pathway within the cell that allows it to uptake glucose. Insulin resistance is caused when a cell requires more and more insulin to uptake the same amount of glucose. One way that can cause insulin resistance is through systemic inflammation or long-term inflammation, which can lead to type 2 diabetes. Now, the reason why type 2 diabetes is so concerning is because it can lead to a variety of complications. For example, it can cause eyesight damage, nerve damage, a stroke, and kidney disease, which is also known as diabetic nephropathy. Diabetic nephropathy is a kidney disease and a leading cause of premature death in people with type 2 diabetes. 40% of people with type 2 diabetes are estimated to go on to develop diabetic nephropathy. Currently, there are no tests that can indicate the risks of developing diabetic nephropathy. One way to create such a test would be to find a biomarker that's associated with the development of diabetic nephropathy. A biomarker is a biological molecule found in blood, other body fluids, or tissues that is a sign of a normal or abnormal process or of a condition or disease. According to the U.S. Food and Drug Administration, a biomarker should be specific for a particular disease, safe and easy to measure, rapid to enable faster diagnosis, cheap, accurate, and consistent. Now, there have been many biomarkers that have been discovered, and some fall under the category of microRNAs. MicroRNAs are short non-coding RNAs that post-transcriptionally regulate gene expression. Here listed on the side are some examples of specific microRNAs that have already been found by previous research to be biomarkers for other diseases. So to sum it all up, the aim of this research is to find a biomarker that indicates the risks of developing diabetic nephropathy, which is a dangerous complication of type 2 diabetes. Such a biomarker, coupled with traditional preventative strategies such as a change in diet and exercise, could help prevent the further development of the disease. As I mentioned earlier, systemic inflammation can cause insulin resistance, which can lead to type 2 diabetes. One way that can cause systemic inflammation are pro-inflammatory monocytes, which are immune cells or a type of white blood cell that promotes inflammation. What can determine if these immune cells or monocytes promote inflammation or not is the amount of methyl groups or methylation on its DNA, which is also known as DNA methylation. DNA methylation and demethylation is the bonding and removal of methyl groups to regulate gene expression. As you can see here, less methyl groups on the promoter region of a gene compared to control generally relates to higher gene expression, and vice versa. If there's more methyl groups on the promoter region of a gene compared to a control, it generally relates to lesser gene expression. And when I say gene expression, I mean the rate at which DNA is being transcribed into RNA and then translated into proteins, which govern the functions within our bodies. So how I went about into finding this biomarker was I received help from the lab that I was working at in first obtaining the IRB approval, participant consent, and the blood draw from an additional 96 individuals to expand on the previously collected 96 samples, making the total sample size of this project 192. I also received help in isolating the monocytes from peripheral blood mononuclear cells. What I did by myself under the supervision of a graduate student was I processed the blood to plasma and blood to peripheral blood mononuclear cells within 24 hours of collection. I extracted DNA and RNA from the monocytes that were isolated from the peripheral blood mononuclear cells and used the DNA from the monocytes to expand on the EPIC array analysis. Based on the results of the EPIC array analysis, I chose one possible biomarker and quantified its concentration by qPCR in monocytes and plasma. Again, the total sample size was 192 and the average age was 32.03 plus or minus 1.4. So because DNA methylation can influence gene expression, which can ultimately influence the concentrations of molecules within a cell, I decided to look at the differences in DNA methylation in monocyte cells between people with and without type 2 diabetes. I then took the results of that experiment and put it into a software analysis, narrowing down my options. Based on the results of the software analysis, I picked one possible biomarker and quantified its concentration by qPCR in monocytes and plasma. So using a form of microarray analysis called epic array analysis, I found that there are 144 differentially methylated genes in monocytes between people with and without type 2 diabetes. The blue you see on the heat map represents hypomethylation, or less methyl groups on the gene compared to a control. And the red you see represents hypermethylation, or more methyl groups on the gene compared to a control. I then took these 144 differentially methylated genes and put them into a software analysis called ingenuity pathway analysis, which relates genes to previously researched pathways or previous literature. Because type 2 diabetes can be caused by systemic inflammation, I decided to look at the genes that were related to inflammatory response, which was 18 of them. So here's a diagram of the 18 genes that were related to inflammatory response by previous research and shown to be differentially methylated in monocytes between people with and without type 2 diabetes. Of the 18 is microRNA 488, the focus of my project. The gene microRNA 488 is hypomethylated in the promoter region of the gene shown by the epic array analysis, which just means that we should expect to see higher gene expression, which should lead to higher concentrations of this gene's product within the cell. Furthermore, another study found that microRNA 488 was linked to the development of diabetic nephropathy in cultured mesangial cells, which are lab-made specialized kidney cells. Therefore, in order to validate that the differences we see in DNA methylation and gene expression actually carry over to the differences in concentration to make for a biomarker, I decided to investigate the concentrations of microRNA 488 in monocytes between people with and without type 2 diabetes. Using a qPCR and a statistical analysis t-test, I found that microRNA 488 concentrations were significantly increased in monocytes of people with type 2 diabetes. Furthermore, there was no expression of microRNA 488 in monocytes of people without type 2 diabetes. This already indicates a possible relationship between microRNA 488 and the further development of type 2 diabetes. However, I wanted to go a step further because the nature of a biomarker is to be quick, easy, and affordable, and I knew that there were quicker and easier ways to extract microRNAs than through monocytes. And one example of that is plasma. So I decided to investigate the concentrations of microRNA 488 in plasma between people with and without type 2 diabetes. Using a qPCR and a statistical analysis t-test, I found that microRNA 488 concentrations were increased in plasma of people with type 2 diabetes. However, the differences were not significant. The results of my research builds upon previous research that linked microRNA 488 to diabetic nephropathy in cultured mesangial cells. It validates that the differences we see in methylation actually carry over to the differences in concentration. And it indicates that microRNA 488 could possibly be used as a biomarker to indicate the risks of developing diabetic nephropathy because microRNA 488 was significantly increased in people with type 2 diabetes and found by previous literature to be linked to the development of diabetic nephropathy. If future experiments holds my hypothesis true, then this would greatly help the Native Hawaiian and Pacific Islander community. Because if we're able to indicate the risks of developing a complication of type 2 diabetes early on, we'll also be able to implement preventative strategies such as a change in diet and exercise early on, and we could prevent the disease from manifesting entirely. Limitations of my research was that I only examined inflammation marker genes, and I focused only on if participants had type 2 diabetes or not. In future experiments, I plan on expanding my sample size to validate my findings. I plan on following up in 2 years to see if any of the participants from this study have developed diabetic nephropathy and related to their current microRNA 488 concentrations. I plan on checking the concentrations of microRNA 488 and monocytes of patients at different stages of type 2 diabetes to further characterize its time course. And lastly, I plan on analyzing which genes microRNA 488 regulates to get a better understanding of its presence. My advice to future researchers would be to just be aware that this is a very time-consuming process, and there's going to be a lot of times where you feel like giving up, and you're going to wonder if staying up and losing sleep is worth it or not. And my advice for you is just to push through those tough times because it'll make you stronger, just like that dog. I would like to thank Mauna Kea Lab, Iolani School, Hawaii Academy of Sciences, and my family for making this research possible. I would also like to thank the participants of this study for giving me their blood. Thank you.